All right, so now we're going to talk about our last cell process, and that is translation. So remember, we're talking about our central dogma of the information in DNA being transcribed into RNA, and then now we're talking about it being translated into our protein. So translation is when we have our RNA then, and our RNA is used to build a polypeptide or a protein. And what do we need? We need 3 types of RNA. So first of all, we need 3 types of RNA. Remember our 3 types of RNA? All right, so mRNA, remember mRNA stands for messenger RNA. And this contains the information about the amino acid sequence. In the protein. Another RNA is our tRNA. Remember that stands for transfer RNA. And this is going to bind the specific amino acid and bring it to the mRNA. So our mRNA contains the information for the particular amino acid. The tRNA is going to transfer that amino acid, meaning it's going to grab onto the amino acid and it's going to bring it to the mRNA depending on the code of the mRNA. Then our last one, our rRNA, remember that stands for ribosomal RNA. And the ribosomal RNA is going to be part of the ribosome, which is where we're going to be using our mRNA to make the protein. It acts as the enzyme to join amino acids together. So we have our mRNA, which is the messenger, which contains the information, the code from the DNA about the specific amino acid sequence. That's going to meet up with the rRNA on the ribosome. So the ribosomal RNA that's part of the ribosome, it's going to act to join the amino acids together that are brought by the tRNA, or the transfer RNA. So then we also need a ribosome. So remember, this is our location of translation. And again, it's made of rRNA, so you have to have that piece of rRNA in there, and proteins, the complex. We have to have 20 different amino acids available. And of course we need to have ATP because again we're building something. This is an anabolic process. We're going to be building this polypeptide chain. So individual amino acids are going to be attached to each other to build the polypeptide chain. So in translation, what we've been talking about is encoding information, right? We've talked about how DNA contains the information for the RNA, and then the RNA contains the information to make the protein. So what does that mean? What that means is, let's take a look here, is we have something called the genetic code. And this is how the mRNA determines which amino acids are in the protein. So how the mRNA determines which amino acids are in the protein. And this genetic code is based on what's called a codon. So a codon is a 3-letter segment of mRNA. So a 3-letter segment of RNA, specifically mRNA. And then each codon stands for one amino acid. And this is red 5' to 3'. So from the 5' end of the mRNA, so that 5' cap, remember we added on the 5' end, so that 5' cap all the way along to the 3' poly A tail. So if we take a look at this, what we're saying then is that we have our nucleotides, right? We have our mRNA, and let's say we have a G, a C, a U, a U, G, G, A, B, C, U, U, C, G, G, and up. So we have a chunk of our mRNA here that we're looking at. Let's pretend our 5' cap is up here. And we're going to read this mRNA. Now, when we read the mRNA, we're going to be looking for our codons, which are 3-letter segments of RNA. So we're going to look at chunks of 3 letters. So this right here, GCU, is a codon, is 3 chunks, or is a chunk made up of 3 nucleotides. Right here, AUG, this is a chunk of 3 nucleotides that make up what's called a codon, a 3-letter segment. This one, GAG, CUU, CGG, and on. So each 3 nucleotides is going to be a codon. Now then, our codons are going to stand for one amino acid. So if we look here, AUG is going to code for a particular amino acid. It always codes for the same amino acid. So let's take a moment and we're going to look at this code. Alright, so what I'm going to do is I'm going to take our mRNA here, I'm going to make a copy of it, and we are going to move it over here where I have some of our code. Here we are. We can move this a little bit, make it a little bit smaller. And we have our mRNA up here with our 5' cap. And somewhere along here with our poly A tail. And then we have our RNA here, our mRNA. So we can see that this is our mRNA. Now what I have brought us to here, this is a very nice table that tells us exactly what these codons stand for. And we read them, again, as these 3 nucleotides, or 3 bases. So if we start right here, we have the first one is the letter A, the second one is U, the third one is G. If we look at this table, we see here this is the first base. So in our case, our first base is going to be the letter A. So we're going to be looking in this row. So these all start with the letter A. Then our second base here is U. So we're going to be looking in this column. So this row, this column gives us this box here that we're going to look in. Then our third base, which is over here, is a G. And you can see that the G is this specific row here. So this brings us to AUG, which is methionine. And the reason I chose this was because this is our start codon. And there's always a start codon, and the start codon is always AUG and always brings methionine. So every single polypeptide, every single protein in the human body is going to start with a methionine because that's how our ribosome knows where to start. So it attaches to this 5' cap, and then it's going to start reading the mRNA in codons, so in every 3 base pairs, until it gets to an AUG. Once it gets to an AUG, then it's going to add a methionine to its strand. We're going to talk specifically about how this happens, but so we have a methionine. Then we look at the next codon. So in this case, we're looking at GAG. So let me erase this kind of mess I made here. So can you figure out which amino acid we're talking about if we're looking at GAG as our codon? So the first base is a G, is an A. That brings us to this square. Third one is a G. So here we are, GAG, glutamic acid. So our second amino acid that will be attached is our glutamic acid, or glutamate. Then let's see if you can figure out the next one, CUU. And you need to be able to do this. I will provide you with this table for the questions, but you need to be able to use the table to figure out which codons you're going to be adding. So CUU, CUU, leucine. And CGG. Let's do one more. So C, G, and G, arginine. All right, so this would be, from what I have written here, our polypeptide chain. And we used codons, and we used our table here to figure out which specific amino acids are going to be attached based on our mRNA code. And again, this is considered, or this is called the genetic code. We have our 3 nucleotides that make up a codon, and then that codon is going to code for a particular amino acid according to this table. So let's look at the specific process then of translation. So similar to transcription, we have our steps. Our first step is initiation. And in initiation, we have our ribosome, our mRNA, and our tRNA are going to come together at the start codon. Do you remember what the start codon is? AUG. And it always codes for methionine. So you should know the start codon. You should be able to read an mRNA strand and tell me where to start and then where to end. If you noticed, I didn't point it out, but if you noticed on our table here, we actually have stop codons as well. If you look up here, if we reach a UAA or a UAG, that's a stop codon. Also a UGA, that's another stop codon. So if we get to those codons, then you're going to stop, and that's the end of our polypeptide. All right, back to initiation. So our ribosome, our mRNA, and our tRNA are going to come together at the start codon. And the way that works is that we have our ribosome. It's actually in 2 chunks. So we have a bottom part of the ribosome, and we have the top part of the ribosome. And in between here, we have our mRNA. And then we have our tRNA that's going to come along. In our ribosome, we actually have 3 spots, kind of 3 seats in the ribosome. They're A, P, and E. So we have these 3 spots here. Our tRNA, our initial tRNA, is going to attach itself here. It looks kind of like a T. It's going to attach itself here in the A spot to our A, U, G. These are our codons on our strand here. And we have next G, A, G. So this is our AUG here, which is our start codon, remember. So our start codon, we have our ribosome. We have our mRNA molecule with our start codon. And then we have our tRNA molecule. And attached to the tRNA molecule, we have the amino acid methionine. So our tRNA molecule is going to come down. I just realized I drew this wonky here. Let me back up. Hang on a moment. Recall, I would like to point this out. that our tRNA molecule is matching up with our codon, which includes the A, the U, and the G. The A, the U, and the G are not in the 3 different spots of the ribosome. They're in one spot right here as a codon, A, U, G, that match up what's called the anticodon on the tRNA. This is our tRNA. And our tRNA has our amino acid attached to it, which is methionine on our start tRNA. So this would be AUG, then we would have, say, GAG, and say UUC. And then we would have others here, right? So let's say UAC, GAU, and on and on. I'm separating these out like this, obviously, to make it very clear that they're codons in chunks of 3. But of course, when we look at our actual mRNA, it's just going to be evenly spaced. Just like, oops, not with T, of course, but A and C and G, just like this. And what we're reading are these 3 at a time. So anyway, we have our initiation, which includes our ribosome, our mRNA, and our initial tRNA, and they're coming together at the start codon, which is the AUG, and is always AUG and always brings methionine. So that's initiation. Then 2, we have elongation. In the process of elongation, we have our enzyme is going to move along the mRNA, adding amino acids. So we should say the enzyme moves along the mRNA, adding amino acids. So in elongation, we have our tRNA here, which is already attached to our AUG, and we have our methionine. Then we have our next codon, right, which is the GAG. If we look back at our chart, our GAG gave us glutamic acid. So what happens then in elongation is that we have a tRNA that has attached to it a glutamic acid. On it, it has the anticodon that's going to be the opposite and match up with GAG. So that's CUC. That's going to come down, and it's going to attach itself here in this next spot. So I'm going to draw that now. So this is going to come down into our next spot in the P location here. And it has glutamic acid on it, so it's attached here. Now, once this tRNA is going to come into this P site on the ribosome, then the enzyme is going to make a peptide bond between the methionine and the glutamic acid. When that happens, it removes the bond here that was attached to this tRNA. So we no longer have that bond. What happens is this tRNA is going to leave this spot, and then what we have is just this bond between the methionine and the glutamic acid. The glutamic acid is still attached to this tRNA that is still bound to the GAG codon. But then this whole ribosome is going to move down, so that now this tRNA that has the glutamic acid attached to it is going to be actually in the A spot. And then what's open next would be the P spot. This would be the E spot here, actually. Anyway, there are 3 spots, and it's going to be shifting down. As it shifts down, the first one is going to leave. That would actually be the E spot, which would be the exit spot. I apologize, I had it backwards, APE. And then we have our methionine bound to our glutamic acid, bound to this tRNA. Then as the ribosome moves down, then we're going to read the next codon, which would be UUC. So if we go up here, we find our UUC. That's going to give us some phenylalanine. So in our process of elongation, then we have coming into our next spot here. Of course, our ribosome has shifted, so it actually looks different now. The whole thing is now over And now we have our 3 different spots here. We're going to have the next tRNA come into this spot, and we said it was phenylalanine. Now when this is attached, then we have an enzyme making a peptide bond between this glutamic acid and the phenylalanine amino acid. So then this tRNA is going to leave just like the other one as this ribosome shifts down again. Once that ribosome shifts down again, then we have another spot here for the next 3, and then we continue. We have another tRNA here that attaches to another amino acid, and then it's going to make another peptide bond, and then we have another tRNA attached to another amino acid making another peptide bond. All the while, once it makes a peptide bond to the next amino acid, then it's going to be released from its tRNA. And then what we end up having, you can see here, is our polypeptide. It's going to continue going on until it reaches step 3, termination. In termination, this is when our enzyme reaches the stop codon. So again, our stop codons are UAA, UAG, and UGA. Once it reaches a stop codon, everything falls apart. These ribosome pieces fall off, the tRNAs are going to be released, and this polypeptide will be released into the cytosol, or into our rough endoplasmic reticulum. So then that gets us at termination our polypeptide chain. So know, though, that not all of the mRNA is going to get translated. So remember, we have these untranslated regions. We have the 5' cap, which is not translated. We have the 3' poly A tail that's untranslated. We have what's called the 5' untranslated region, or 5' untranslated region. And we have the 3' untranslated region. And these do not code for amino acids. These are just the chunks before and the chunks after. Remember then that we have this 5' cap, and then we have some mRNA before we're going to reach the AUG, which is the start codon. So this is going to be our untranslated region, our 5' untranslated region. And then we're going to have our let's say lots and lots and lots of codons here that are going to make the bulk of our mRNA. And then that's going to be our coding region. And then we have over here, before we reach our poly A tail, we have the 3' untranslated region. And again, we have regions like this for protection. We don't want to get to our important information. If something's going to be eating away at the ends of our mRNA inside of our cell, because we do have enzymes that eat or break apart mRNA and DNA, that's for protection, so we can break apart the DNA or mRNA of bacteria, for example, if we're trying to break them down and make sure that they can't make their own mRNA or make their own proteins. We have these different kind of things put in place, like the modification that we saw earlier with the 5' cap and 3' tail, and then these untranslated regions. So the last thing that I want to talk about in cell processes is something called gene regulation. And gene regulation is basically determining if a protein is made or not. And we have different spots where we can have gene regulation. So many points of regulation. So the first one is transcriptional regulation. So transcriptional regulation is determining if the RNA is going to even be transcribed. So this is based on the unwrapping of the DNA from the histones. So unwrapping of DNA from the histones. Remember, the histones are those proteins that we wrap the DNA around to get it really condensed so we can move those things around. And also proteins that are binding to the promoter to stop or to help RNA polymerase from binding. So remember, we have that promoter region. And we can have proteins binding. So proteins binding to it to help or stop the binding of the RNA polymerase. And this way we can turn on genes and turn off genes by the attachment of different proteins. Another spot where we have gene regulation is called post-transcriptional regulation. So post-transcriptional regulation. In post-transcriptional regulation, this is where we control the processing of RNA into the mature RNA. So we control processing of RNA into our mature RNA, mature mRNA. So this would consist of adding more adenines to the poly A tail so we can slow down the degradation, so if anything's eating at it, so that more protein is made, so we can add adding more A's to the 3' poly A tail, so we can slow degradation. And if we're slowing degradation, then we're increasing the proteins made. We can make more proteins the longer it sticks around. And then also we have splicing, and splicing can control which forms of a protein are made. Third spot where we can see gene regulation is translational. So translational regulation. And so this is going to determine if the mRNA will be translated into a protein. So this is going to affect the formation of the initiation complex at the beginning of translation. So the initiation complex. So remember, we have the initiation region, and then if we change, so if we have this initiation complex and we change the formation, so if we don't form that initiation complex, then we're not going to see that mRNA be translated into a protein. Also, our 5' untranslated region is going to help to regulate initiation. So again, kind of turning it on, turning it off. We're not going to get into too much detail. It's just important that you know that there are these 4 different areas that we can decide whether or not we're going to continue on with this process. So the fourth one is post-translational regulation. So this is where we're going to control the folding, the activation, and the breakdown of the protein. So it controls the folding, the activation, and the breakdown of the protein. So some proteins last a long, long time. So we have them hang around for a while. So sometime even something like 90 years. Other proteins degrade quickly. So think of some some hormones, things that we want to have that message go, and then we want to degrade it. We don't want to keep sending that message. All right, so those are different types of gene regulation. So determining if we're going to get from that DNA all the way to the protein. So we have transcriptional regulation, so regulating at the transcriptional level. We have post-transcriptional, so right after we've transcribed it, we can still regulate it. We can still make it so that protein never gets made. And then we have translational regulation, so we can regulate it whether or not it gets made at that point. And then we even have post-translational regulation. We can regulate it even after we've made the polypeptide. All right, so that summarizes our cell processes. We ended up here with our translation. So remember, we were talking about the process of going from DNA to RNA to a protein. We talked about transcription, going from DNA to RNA, and now translation, going from RNA to a protein. We talked about our genetic code, understanding the codon. You need to know how that works, the three parts. The codon are the three letters, and then how to read a table like this to get our amino acid strand. You should know the three steps: initiation, elongation, and termination, and how that works. And then you should understand gene regulation that we just talked about, the different ways that we can ensure that something gets made or stop something from getting made.